Okay. Good morning, everyone. Good afternoon. Already afternoon here. My head is not working very well. So thanks for coming to the seminar. So I'm very glad to invite Dr. Adrienne Gorney to give a seminar here today. So Henry is currently an assistant professor at NC State, work on the provider nematology over there, but she actually got her bachelor degree in botany in our department a while ago. Then she moved to the Cornell, got her PhD, and from there she got the faculty position at NC State. So at NC State, I think she mostly focused on the root knot nematodes, especially there's one very aggressive emerging called the guava root knot nematodes, problematic for the vegetable, and especially sweet potatoes. I think she will talk more about that today. Yeah, with that, it's all yours.
Awesome. Yeah, well, thank you so much for the invitation to speak with you here today, and I'm really glad to come back to a home department. So yeah, today I want to present a bit about research and extension nematology in North Carolina. So an outline of today's talk, I'll give you a snapshot of what agriculture is like in North Carolina. There's some key differences, I think. And then we'll get into opening this can of worms and talking about Meloidegein enterolobii, which is the primary root knot nematode species that I work with. So we'll kind of talk about that initial identification and its significant impacts on sweet potato production in North Carolina. And then I'd like to share with you just a snapshot of the few major research programs I have going on in my lab. Not a complete list, but just a summary. And then finish up with a summary and looking forward. But first, just a very brief introduction to kind of follow on what Lei gave. I'm originally from Canton, Michigan, about 30 minutes outside of Detroit. And it was actually my high school AP biology teacher who got me interested in botany and plant science. And so from there, I applied to Purdue for an undergraduate and came to this department. I've actually been to many seminars in this room. I never thought that I would be giving a seminar in this room, but it was here at Purdue that I continued this interest in plant science research and was also exposed to extension work. And through a summer research internship at Cornell, kind of confirmed that interest to go on to graduate school and continue with extension. So I did my PhD at Cornell University in the lab of Sarah Pethybridge. We were based at the agricultural station in Geneva, New York. That's about an hour north of the main campus in Ithaca. And it was about 6 months to my graduation when basically my dream job at North Carolina was advertised of a research and extension nematology position. I hadn't yet graduated. I didn't think I was suitable yet for the position, but many people urged me to just send in an application, go for it. And lo and behold, they interviewed me and apparently liked me. So I started at NC State in 2019 as an assistant professor. At NC State, I have a 60% extension, 40% research appointment. And on paper, my commodity responsibilities are sweet potato, soybean, cotton, corn, and tobacco. But being the only applied nematologist right now at NC State, I also help with questions and some research in peanuts, vegetables, sometimes even ornamentals. So it's enough to keep me busy. So at NC State, I run the plant nematology lab. And our mission is quite simple, is to conduct applied research into biology, epidemiology, and management of plant parasitic nematodes that are of economic importance in North Carolina. But we do have a heavy focus on the root knot nematodes, the Meloidegein species. Our overarching goal in our research and extension is to provide informed nematode management decisions through our research. And this is just a snapshot of some of the team over the years. So just a brief background of what agricultural is like in North Carolina. It's very big. We have over 50,000 farms, many of them family farms, working over 8 million acres and contributing to a $92 billion industry. It's also extremely diverse. We have nearly 80 different crops and commodities that are grown from the coast to the mountains. In North Carolina, we have very distinct growing regions. On the eastern side, we have the coastal plains region, characterized by very sandy soils. In the central part of the state, we have the Piedmont. This tends to be more clay soils. And then the mountains in the western part, this is where most of the tomato and vegetable production is located. North Carolina claim to fame is that we are the top producer of sweet potatoes in the country. Nearly 60% of the US sweet potato crop is grown in North Carolina. We also lead the US in flue-cured tobacco production, and we're second in Christmas tree production. So agriculture is, needless to say, a very big economic driver in the state. Because of that, North Carolina has very much a robust extension system. We're one of the few states that still maintains a county-based extension system. So in all 100 counties and with the Eastern Band of Cherokee, we have extension agents and extension offices within that. That means a team of over 1,000 extension agents, faculty, and staff based in Raleigh, research stations, and these offices throughout the state are working towards these goals. And of course, our point of pride is the sweet potato production in the state. A bit about sweet potatoes. You may have hopefully have eaten a sweet potato before, but sweet potatoes are not the same thing as what we might call white potatoes or Irish potatoes, and they're not the same thing as yams. True sweet potatoes are in the morning glory family, whereas white potatoes, of course, is a solanum species in the nightshade family. True yams are in a completely different plant family. And just to illustrate, this is a sweet potato here and a yam on the left. In North Carolina, sweet potatoes are typically planted in the May to June timeframe and then typically harvest in the September-October timeframe, really trying to hit that Thanksgiving market. The counties that produce sweet potato are really diverse and spread out across the state, but the vast majority of production is going to be centered here in the central coastal plains region. The majority of the sweet potatoes are going to be for the fresh market, so these might be things you buy in the grocery store, but we've also had an increase in canning and other value-added products, such as sweet potato fries, which are actually my personal favorite. An important note here is that approximately 40% of the entire North Carolina sweet potato crop is actually exported, mainly to the European Union, but also to Canada and elsewhere. And that's actually going to become important later in the talk. I wanted to go through just exactly how sweet potatoes are grown because it's a very different system. And when I arrived at NC State, I had to kind of learn this, and it's a fascinating system, really. So very early in the season, foundational seed crop producers lay out seed potatoes in these very dense beds running up the field. As you can see, they're really packed in there. They will then cover these beds with either plastic or a thin layer of soil, and that heats up the soil and causes the roots to sprout, and they grow into these very dense plantings of the sweet potato vines. Once those vines come out, workers will come through the field and they will cut the vines at the soil line and cut them into approximately a 10 to 14-inch segments. These are called slips, and these slips are then sold to other farmers who take them and plant them in their field using these mechanical setters. It's actually quite astonishing that they just plant these short segments of the sweet potato. They rapidly grow roots and start to spread out and vine and eventually grow the sweet potato storage fruit. Sweet potato, it's about a 100 to 100 day crop, so quite quick. And they produce these very pretty flowers, reminiscent of the morning glories. Once the crop is ready to harvest, they will come through with tractors and turn over the soil, exposing the roots then to the surface. All of the fresh market sweet potatoes are going to be hand harvested by people, workers coming through the field and picking them up by hand. That's because the sweet potato skin is actually quite sensitive and susceptible to skinning damage. So we want to minimize that damage during the harvest process, both for cosmetic reasons and to reduce the risk of secondary pathogens moving in after harvest. And I have to say, the speed and dexterity with which these farm crews work is astonishing. They'll harvest this whole field in a half hour. Once those sweet potato roots are picked up, they're put into these wooden boxes, which we call bins, and they have to undergo a curing process. So the sweet potatoes are held at about 85 degrees Fahrenheit at high humidity for about 3 to 5 days. That sets the skin, hardens the skin, and allows them to be stored for a long time. They can be stored for up to 13 months at about 55 degrees Fahrenheit, again at high humidity, in these vast, giant warehouses that are centralized throughout the region. They stack them up really high, and those bins are going to be labeled with codes for the particular cultivar of the potato, the harvest date, the farm from which it came, and sometimes even the field location from which it came. So once they come out of storage, they're going to be moved to a packing facility. So they're moved to the packing facility, they're washed free of soil, they're graded out for size and shape, and then prepared for delivery. So here on the left, these are individually packaged for a supermarket with individual potatoes. These on the right are what we call 40-pound restaurant boxes that might be sold to a restaurant or other food prep facility. These particular ones are actually a purple fleshed sweet potato variety that's really popular in a lot of Asian cuisines. So that was quite interesting. So this whole process of planting them in the field, cutting the slips, allowing them to grow, harvesting them, you might start to see some red flags of where plant disease and plant pathogens can move in and can spread during this whole process. And that's kind of where the next part of this story goes, that when you open the can of worms, an opportunity emerges. In North Carolina, we are blessed or perhaps cursed, depending on how you look at it, with a variety of different plant parasitic nematodes. We have a very diverse array. We have root knot nematodes, soybean cyst and tobacco cyst, and lesion nematode are some of the more widespread nematodes. But we'll also see sting, stubby root, and Columbia lance nematode in pockets throughout the state. But chief among these is really the root knot nematodes. We have the 4 major species of root knot nematode: Moedigine incognita, Hapla, arenaria, and javanica. However, recently, we've had the introduction of a new species of root knot nematode. So back in 2011, very severely galled cotton and soybean plants were sent into the Department of Agriculture. These were a bit unusual because the galling was quite severe. It was initially thought to be Moedigine incognita, that's the southern root knot nematode, because this one's very common in the state. But there was something about the severity of the symptoms that prompted the diagnosticians to look a little bit closer and to do some sequencing. And sequencing revealed it to be Moedigine enterlobi, commonly known as the guava root knot nematode. This was a new species in the state and of high importance, mainly because enterlobi is considered highly pathogenic and has a very broad host range. As you can see here, it produces very severe galling symptoms on the sweet potato storage roots itself, and also on the feeder roots or the fibrous roots that feed that storage root. So those potatoes are a complete loss. They are unmarketable. Furthermore, in North Carolina, sweet potato is often grown in rotation with tobacco and soybean, and sometimes cotton and other vegetables. Unfortunately, Enterlobi is also feeds upon these rotational crops as well. And we've seen that it loves to feed on tobacco. So malignant Enterlobi, it's not necessarily a new species. It was first characterized in China in the 1980s. It has since been identified in several countries throughout the world. It got its common name, the guava root knot nematode, because it caused very severe epidemics in guava orchards in Brazil. In the US, it was first identified in Puerto Rico, but in the continental US, it has been identified in Florida in 2001. Of course, the 2011 report in North Carolina. And since then, it has also been identified in South Carolina, Georgia, and Louisiana. Within North Carolina, we are doing active sampling and monitoring to look at the spread of this nematode. And those counties that have had positive reports of the nematode are highlighted here on the map. And if you can think back to that map of where sweet potato production occurs in the state, we see a lot of overlaps between that map and this one here. Just as a note, we do aggregate this to the county level so as not to out one particular farm or another. So even if one field has been confirmed positive in the county, we count the whole county as positive. So the first report of Enterlobi in the state came out, and this is of concern because Enterlobi is on the EPPO A2 pest quarantine list. This is important because remember, we're exporting about 40% of the sweet potatoes to Europe. So regulatory agencies and inspectors in Europe are monitoring for this nematode, and there's a zero tolerance for it. So this is why it's of great concern to our sweet potato growers. Because of that, the North Carolina Department of Ag quickly issued alerts and information about the nematode and its impact on sweet potato production. And NC State, in cooperation with extension partners, began to offer some management guidance and recommendations. This is actually about the time that I came into NC State in 2019. And when I came into NC State, I first noticed that there was still a lack of information. A lot of growers and their farm workers had questions about what exactly is this nematode? What are we supposed to be looking for? We've heard about it, but we don't know what to do or even what to look for. So one of the first things we did was to develop a training program for the farm laborers. And this was to support immediate education and awareness. In collaboration with the Extension Farm Worker and Health and Safety Program, we developed a bilingual training program that uses these posters and pictures of infected sweet potatoes to show them what to look for in a field or packing house setting. So the idea here is to inform the workers what infection might look like so that they can then alert supervisors or the farm owner so they may quarantine that field or take precautions to limit its spread. So this was the initial fire to put out, but we had to start then developing some research to how to better manage this nematode. And many of you are perhaps familiar with the Integrated Pest Management, or IPM. And I took this definition straight from the dictionary, as IPM is an attempt to use all the available methods, biological, chemical, cultural, genetic, legal, and physical, to control the diseases and pests of a crop plant for the best control of results, but with the least cost and least damage to the environment. In the case of nematodes, often prevention is the best medicine, though. If we can prevent the nematodes from establishing in the field, we can keep fields highly productive and clean. So this definition is good, it's accurate, but it's a little bit long and stuffy. I don't know if we have any fans of Captain Planet or other like 90s TV shows. I am, both objectively and because I think it actually really well illustrates an IPM approach. So we have to use things by our powers combined of cultural practices, chemical management, host genetic resistance where available, and continue expanding our knowledge of the biology, epidemiology, and distribution of this nematode to develop a comprehensive, holistic plan for nematode management. So that's where the rest of this talk is going to go. I just want to highlight some of the active research projects in the lab within each of these categories. So first, starting with chemical management as a short-term, immediate stopgap measure to this nematode. So as part of the extension component of my lab, we often trial and test different nematicide compounds and combinations in the field to look at which ones may be best for management of enterolobi. And I wanted to highlight what one of these trials actually looks like. So we performed this nematicide trial on farm in collaboration with the grower cooperator near Princeton, North Carolina. And we were looking at both fumigant and non-fumigant nematicides. So one very popular fumigant nematicide is telone-2. This is 1,3-dichloropropene, very commonly used in sweet potato and tobacco production. So we included that one. We also included this newer nematicide called Celebro. This is fluazza-indolazine. It's a new one to the market, recently registered for sweet potato. So we included that. We also looked at combinations of both the fumigant and Celebro at sort of full rate and the half rate here. And we also had non-treated plots to compare them to. So we put out the products in a small plot replicated trial, made the applications, planted the sweet potatoes, and came back and harvested. I should mention that we did this with Covington sweet potato. I didn't mention it previously, but Covington is our top variety. About 90% of all the sweet potatoes in North Carolina are Covington. So it's almost like a monoculture situation. And then in this trial, we collected quite a bit of data. So we looked at storage root yield. We looked at the incidence and severity of galling damage, quantified the yield, the marketable yield, and we also collected soil samples both at planting and at harvest to look at how these nematode populations are fluctuating under each of those treatments. We found quite stark results in this particular trial. So the table here on the left is just the numeric results of each of the treatments. The non-treated is the last line here, and the different yield metrics. We did have a significant difference in terms of marketable yield. So we're calling marketable yield clean, not galled, #1 size potatoes. So that's something about that size. The bar graph here on the right is that marketable yield under each of the treatments. And what we saw is that we had very, very low yield in the non-treated and significantly higher yield under each of our treatments, with the telone plus Celebro giving numerically the highest marketable yield. This trial was exceedingly hot. We had very high nematode populations and also quite significant deer damage. These numbers, these yield numbers, are not typical of what we would see in a commercial setting, mainly because of that deer damage. Looking at some of the other metrics that we collected, the graph on the left is for the incidence of galling. So out of the #1 size potatoes, how many displayed galling and how many were clean? We had the highest incidence of galling under the non-treated plots at about 75% of the potatoes galled. And each of the treatments had less incidence of galling, although that relationship was not significantly different. In terms of nematode populations, there wasn't much rhyme or reason to the harvest counts. And we didn't see any significant trend among the nematode populations at harvest under each of these treatments. This is actually quite typical in what we tend to see in these trials. Our nematicides are very good at knocking down early season nematode numbers to allow that crop to establish. But by the end of the season, we see a significant rebound in nematode populations. So there's really no carryover effect of a fumigant or non-fumigant nematicide to the following year. So these nematicides are good for, again, a short-term management method, but we're seeing that they are not a long-term or sustainable answer. Just visually of what some of these treatments look like. Just some examples from the plots. This is a non-treated plot, very few sized potatoes, and it's difficult to see, but there's a lot of galling on these roots. Within our other treatment plots, we see much larger potatoes, and they're overall far more clean in the sense of less galling. So these nematicide trials are something that we do every year at multiple locations in different crops and commodities. Our idea is that we want to provide updated, accurate information to the growers so that they can then make their own decisions about whether or not to apply nematicide and which one to apply. So again, this is a summary of all of the different nematicides that we've tested over the years and relative level of efficacy for enterolobi. I will note that the fumigant Telone-2, it's quite expensive. It's over $300 an acre to fumigate with this product. So it's not very economically sustainable, and many growers are looking for alternatives to using this fumigant product. Solibro is cheaper, but not much better at about $100 an acre to use this product. So again, short-term stopgap measures, but we need to look at longer-term management tactics. So some additional thoughts that I just wanted to include about these nematicide trials. If you've ever worked with nematodes, they can sometimes be a pain in the butt. You go out and you do your pre-plant sampling to figure out where to put these trials. And sometimes you'll see very high pre-plant samples, but then when you go to harvest your trial and evaluate the data, you see very low incidence of damage, which doesn't make much sense. Or vice versa. We sometimes do our pre-plant sampling, find moderate populations, but then we see very high incidence of damage at the end of the trial. So these relationships are often influenced by soil type and environmental conditions during the growing season and can be quite aggravating. Additionally, the spatial variability of the populations. Nematodes are known for being spatially variable at both a within field or even a hyperlocal level. So accounting for that variation in how we set up and conduct the trial is important. We also do see mixed populations, and in some fields, it's common to have both incognita and enterolobi present in the field, which may impact how well a particular product performs or a particular rotational crop. And through all of this, I don't think we can forget the human connection of all of this. The highly pathogenic nature of this nematode, coupled with those very expensive nematicide options, and there's a regulatory aspect to this. This is having a significant impact on a lot of livelihoods. And we can't forget that behind all of these decisions, there are real people, real families, and real farmers making these decisions. Okay, moving on to just a snapshot of the next project, which is looking at different cultural practices on the farm for nematode management. And one cultural practice that we can use is actually crop rotation. And we do know that Moidigai enterolobi, peanuts and corn seem to be good non-host to this nematode. So the nematode doesn't feed or reproduce well on these crops. However, we do know that tobacco and soybean are very good hosts. So we were interested in conducting a trial to see what a multi-year rotation can have for managing enterlobi in a following sweet potato crop. And this trial was set up and managed by a really bright master's student, Baker Stickley, who we've been working with. So what we did is, in collaboration with a local grower cooperator, we selected a field that had high enterlobi counts, and the farmer helped us plant out these long strip plots of our different rotational crops. So we had peanut, soybean, tobacco, and corn running up the length of the field. So that was in the first year of the project. Once those main rotational crops were harvested in the fall, we came back through and planted perpendicular different cover crops. Cover crops are pretty popular in North Carolina. It's very common to use something like wheat to plant after a corn or a sweet potato crop. So the farmer helped us plant these cover crops going in the opposite direction here, or perpendicular direction, replicated up the entire field. We had 4 replications, but I could only fit 2 on a slide. So this was in the fall of 2023. Then in the spring, we came back through and applied a fumigant over half of the area. So you can see a pattern developing here that we had different combinations of main crop, cover crop, and fumigant application replicated throughout the field. It was not randomized, but we had to set this up in a way that worked with the grower's equipment and worked for our sampling strategy. So then after this, in the spring of the second year, in the summer of the second year, we planted sweet potatoes over the entire area. And we came back through and we collected then data on the yield, sizing them out, sorting for incidence of galling, and also collected soil samples for the nematode counts. And I'll walk through the initial data we found. So these are the nematode sampling data from the fall of 2022. This is immediately after those main rotational crops, and the average of the nematode counts under each of the main rotational crops. And what we saw was that there was a significantly higher population of root knot nematode, enterlobi, following the tobacco crop as compared to soybean, peanut, and corn, whereas peanut had the lowest population counts. This actually fit quite well with our hypothesis, as we knew that peanut was a non-host to enterlobi, whereas tobacco was a very good host. Personally, I was expecting the corn to perform a little bit better than it was here. But again, I think this may be because of a mixed population of both enterlobi and incognita, which is known to feed on corn. We're going to be doing some following up sample and species identification this year to confirm that. Next, we performed soil sampling right before the fumigant went out to see if there was any carryover effect from that rotational crop to the following spring. Long story short, there was actually no statistical difference between the main crops and even broken out by cover crops under each of these segments. So it appears that the populations were more or less evened out over the winter. We then came through, and in the summer of the following year, we planted the sweet potatoes and harvested them and evaluated the yield. I'm not presenting all of the yield data just for time's sake, but here is the marketable #1 size roots by the cover crop in the fall of 2023. So again, marketable would be a #1 size potato with no galls. And what we saw was that under the tobacco, regardless of a cover crop, there was significantly lower, well, not in all cases, but numerically lower marketable yield following this tobacco section than any of our other corn, peanut, or soybean areas. And it was actually quite stark in the field. You could literally walk through and see the galls in the potatoes following the tobacco crop and not see many galls in potatoes following corn or peanuts. Interestingly, there didn't seem to be a significant trend of the cover crops, either the black oats, wheat, or a fallow section as control under each of these sections. We're not really quite sure why this is. I was expecting a response of the cover crop for this. It may be because we had a very dry, warm year. The cover crop didn't grow that well, but we're going to continue to look at that. We can even look at this now with the fumigant application laid on top. And this is a bit busy, but again, we have our main rotational crops, corn, peanut, soybean, and tobacco. The cover crops are the different colors, and then the lighter bars are the fumigant, the darker colored bars are the no fumigant applications. Again, we saw a trend in that even with the fumigant application, did not save essentially the tobacco areas, in that we still had lower marketable yield following tobacco, irregardless of the fumigant application, when compared to these other areas. It was interesting in that both the corn and the peanut sections, there was a numeric increase in marketable yield following the fumigant as compared to no fumigant, although those relationships were not statistically significantly different in these cases. So our key takeaways, and something that we can provide immediate information back to the grower, was that nematode populations for Enterolobi were highest immediately following a tobacco planting compared to corn, peanut, or soybean. Our marketable yield of sweet potato was lowest within that tobacco group. And surprisingly, cover crop did not appear to have a significant effect on the nematode populations, at least with that pre-fumigant sampling. We did see a numeric increase in that marketable yield within the fumigant, within the corn and peanut group. But we are actually repeating this trial with 3 additional growers over multiple locations so that we can gain a larger data set. But this is information we've presented at extension events throughout the state, and many growers are now interested in learning more about how they may incorporate these non-host crops to increase yield in a following sweet potato crop, as that is their most profitable crop. All right, moving right along to another research project, which is host genetic resistance. So one thing is that Maloidiga enterolobi is known to overcome host genetic resistance that is effective in controlling other root knot nematode species, such as the MI gene in tomato or the RK gene in tobacco, also in soybean and cotton. So this is of concern as we are not able to deploy these resistant varieties in the field to manage this nematode. So we really need to look for new sources of resistance that we can then work with plant breeders to characterize and incorporate into their new materials. So one way we do this is through greenhouse bioassay screenings. And we do a lot of these, but this is one project that was led by a PhD student in the lab, Tanner Schwartz, who has since graduated. But what Tanner was interested in was screening a variety of different commercial and wild soybean varieties for potential resistance to Maloidiga enterolobi to begin characterizing this new source of resistance. So Tanner selected about 44 commercial soybean varieties and 77 wild soybean varieties, planted them out in the greenhouse, allowed them to germinate, and then systematically inoculated them with a even level of the nematode across all of the replicates. We usually include tomato in these trials as a known positive control. He allowed the plants to grow, and about 60 days later, he came back, uprooted the plants, and looked at the roots. We score these on severity of galling, and then we perform an egg extraction and count the nematode eggs. We use that as a proxy for how resistant or susceptible that particular line was. So for this, we had 6 replicates per accession. We inoculated them with about 2,500 eggs per plant, which is quite a large amount. We really want to put these plants through their paces, and evaluated this through 2 independent trials. So these greenhouse bioassays, they're really the gold standard for evaluating resistance in a line to nematodes, but they can be very laborious. It takes a lot of time and a lot of training to perform a trial like this, but we usually get very good data at the end. So what Tanner found, and this is a bit busy, but essentially what he found was that none of the commercial soybean varieties were resistant to enterolobi. That was information we could provide immediately back to the growers and say that these varieties you're planting in your field are not resistant. We need to look at other management methods. However, what was exciting was that a whole 5 out of the 77 wild soybean varieties did display resistance and strong resistance to enterolobi. And those are highlighted up here. So this is information we were then able to provide back to our plant breeding collaborators, and he's able to make crosses for us, and we're hoping actually to evaluate a new set of crosses from him next year. This is work that a new student, Sarah Cates, will be continuing on, and this was recently published as well. So again, these trials are really the gold standard for providing this resistance data, but it can be very, very laborious. Just to show you what this actually looks like when the trial is done, these are just some examples of the different soybean lines. So we have wild soybean here on the left, and some of the cultivated soybean, commercial soybean on the right here. This particular line was highly resistant, very clean. It was hard to find a gall on that root, whereas in the susceptible, it is extremely susceptible, very heavy galling within these, and the commercial soybean as well. So these trials, we have to look specifically at each root, rate the severity of galling, and then perform the egg extractions and counting. The life of a nematologist. Okay. So the last research section I wanted to touch on is these more fundamental studies in looking at biology, epidemiology, and distribution of nematodes in North Carolina, specifically enterolobi. And within this project, one recent survey that we did was looking for plant parasitic nematodes and enterolobi in the tobacco producing regions in North Carolina. And this project was headed up by master student Anna Bonyak, who has also graduated. But we know that enterolobi reproduces rapidly on tobacco, and nearly all of the flue-cured tobacco in North Carolina contains the RK resistance gene that is effective in controlling incognita race 1 and 3, and also arenaria race 1. But it does not appear effective at managing this enterolobi. So we wanted to gain more knowledge about the distribution of plant parasitic nematodes and the distribution of enterolobi in the state. This was really driven by the fact that a survey like this hadn't been done in nearly 40 years in the state. So there was a significant knowledge gap. And so we really accomplished this with the help of extension agents throughout the state, who sampled 14 counties in 2021 and 10 counties in 22. They collected the samples and submitted them to the North Carolina Department of Ag Nematode Assay Lab, who evaluated and counted the samples for us. We also collected information on the GPS coordinates of the exact location from which those came, and also information about the particular tobacco variety that was grown and the previous rotational crop that was prior to that tobacco crop. And the counties that were sampled are highlighted here for each year. So what Hannah found in this survey is summarized briefly here. So on the left, we have the results from the 2021 survey. We had 108 total samples, and these numbers reflect the frequency of different nematode genera that were found. So for the Pratolynchus, the lesion nematodes, these were found relatively frequent at about 34%. Root knot was also very prevalent. Dagger and stunt were also quite prevalent. In 21, we only had one county with report of tobacco cyst nematode. And that's actually significant. Tobacco cyst nematode, Bovidera tobacco, can be highly damaging to tobacco and very yield limiting. Traditionally, this has been limited to a few counties in the north bordering Virginia. So it was actually great to see that this one was not occurring in any of our other sampled counties. So it seems to be still limited to those counties bordering Virginia. So the growers are doing a good job at keeping this one at bay. Out of the root knot nematode samples, out of the samples that had root knot nematode in 21, only Incognita and Hapla were found. We didn't find any enterolobi in our survey in 21, which was a little bit frustrating because we set up this whole survey specifically to look for enterolobi, and we didn't find it, which may actually be a good thing. In 2022, it was actually a similar story. So we had 70 samples total in 22. Lesion, root knot, dagger, and stunt were also very common. We did not have any reports of tobacco cyst nematode in 22. Of the samples that had root knot nematode, Incognito was the most common, and we did find 2 samples that came back positive for enterolobi, including a new county in the north of North Carolina, which was a first report for this county. So this provided some excellent information just to get a sense of what nematodes are most frequent, what appear to be most common in our tobacco growing regions. And we were able to further track, at least in the case of the 22 samples, where enterolobi was occurring. We also looked at the density of nematode counts from these samples. And just for brevity, I'm just displaying the results from 22. So this table summarizes the population counts for each of the major genera under each of the counties that were sampled. So immediately we saw here that root knot continues to be at very high density almost across all of the counties that were sampled. This tells me at least that our research and extension efforts into root knot nematode continue to be justified in this case. However, lesion nematode, stunt, and in some cases soybean cyst was also starting to get into ranges that might be considered above threshold or problematic. We may want to keep an eye on these particular nematodes moving forward, looking at further management and research into them. Yeah, I think that's all I want to say here. We also collected data on the tobacco variety and the previous crop that was grown, and we were able to perform some correlation statistics to see if there was any effect of the tobacco variety on the density of the nematodes. So the table at the top is for the 21 data. The table at the bottom is for the 22 data. So, and then these are the averages of the particular nematodes that corresponded to each of the tobacco varieties that were recorded. Long story short, there was no significant effect of the tobacco variety on the densities of nematodes that were found, either for root knot or any of the other reported nematodes. This is not wholly unsurprising, as we do know that all of the commercial tobacco varieties have that resistance gene, which is effective against certain species and races of root knot, but not effective at controlling these other nematodes. The story was a little bit different for the previous crop. So again, the 21 data is in the table at the top. The 22 data is the table at the bottom. So we did have a significant effect of previous crop on root knot nematodes, dagger nematodes, and spiral nematodes in 2021. For the root knot nematodes, our populations were highest in tobacco following sweet potato, which again is a very common rotation in North Carolina. In 2022, it was a different trend that we saw, in the sense that we only lesion and stubby root nematode were significant. For lesion nematode, we saw the highest populations following wheat rather than tobacco behind tobacco, sweet potato, soybean, or cotton or corn. So this is adding to our knowledge base of how these rotational crops may affect nematode populations, and how we might start looking at leveraging these rotations for better nematode control. It is surprising that we didn't see similar trends between years, but we're actually going to do some follow-up soil sampling and continue on this particular project. All right, if I'm doing okay on time, I'll just wrap up with a summary of kind of future directions. I wasn't able to touch on all of the different projects we have going on in the lab, but I'll just briefly summarize them here. A major pillar of the lab is continued resistance evaluation and screening, working with plant breeders to identify new sources of resistance, try to characterize that resistance, looking for loci associated with that resistance. We have active projects going with the sweet potato breeder, Dr. Craig Yencho at NC State, in cotton with Martin Rubin, USDA ARS, soybean and vegetables. This is led by graduate student Sarah, also previously Ashley Philbrick, master student who graduated, and Marcelo Chavez, who made significant contributions. Simon Frere is a PhD student with Craig, who is really the leader in the sweet potato work. We've also had some excellent undergraduate researchers contribute significantly to this pillar, including Kelsey and Grace. Another big pillar of research that I'm highly interested in, but I didn't have time to really present today, is using new molecular tools to enhance nematode diagnostics. Right now, whenever growers send in a soil sample to the lab, live nematodes have to be extracted, identified, and counted from that sample. It requires a lot of training and a lot of experience to do an accurate count. But I'm interested in how we can use molecular tools to get rapid, robust species-level information and counts for these nematodes. And we have a postdoctoral research associate working with us on this, and this will also be a component of Sarah's project. We're going to continue on with our survey work with another survey affecting vegetables. Again, this was driven by the fact that a survey in vegetables also hasn't been conducted in NC State or in North Carolina in over 40 years. And this is one that we're doing with our collaborator out in the mountains, Inga Meadows. We, of course, are going to continue supporting our extension objectives to provide continuous nematicide efficacy data. As new products become available, new biological materials, or new application methods, we want to be the first to test those and provide unbiased recommendations to the growers. And this is led up by the two awesome research technicians in the lab, Bennett Jeffries and Jessica Dautre. We're also continuing our cultural practices evaluations, that rotational crop trial, and also doing some sanitation studies for the packing house and storage facilities. However, all of this is really driven through the input, support, collaboration, and funding from commodity groups, the grower cooperators, our industry partners, and the extension agents who are seeing what things are like in the fields in their own counties, with our ultimate goal of supporting these informed nematode management decisions. So with that, I'll just say thank you very much for listening to my seminar. I want to say a special thank you to several researchers at NC State, including Dr. Nina Quesada-Ocampo, Craig Yencho, Jonathan Schulteis, and Matthew Van, who are some of our key collaborators. This is my lab group up here. We're a small but mighty group. And when I was putting together this presentation, I was digging through some old photos, and this is the Purdue Botany Club circa 2014 when I was in it. So yeah, and thanks. I want to acknowledge the funding sources as well. To USDA NIFA SCRI grants and support from the North Carolina Sweet Potato Commission, Soybean Growers Association, Cotton Incorporated, and Corn Growers Association. So if there's any questions, I'd be happy to try and answer those.
Why sweet potato need such a high humidity in storage?
Storage, it has to do with the transpiration of the sweet potato. They very easily lose water and shrink. So that's to help prevent that shrinkage and that loss.
Would that have mold issue or?
The mold issues,
yeah.
They do every so often. They do. Insects, a lot of fruit flies can be problematic, some mold issues. So ensuring that there's as minimal skinning damage as possible is important to prevent that.
I noticed on one of the early slides where you talked about how we showed data on marketable, like a drop in the marketable quantity, but then there were other categories. Can you eat them if they have the lesions?
That's a great question. So can we sell the potatoes even if they might have lesions? Growers could sell them to a canning facility or a French fry maker, or we actually recently had a dog food manufacturing company open in North Carolina, and they are making a particular line of dog treats that are made out of sweet potato. So we've had some growers sell damaged potatoes to them.
Yeah.
But of course they're getting pennies on the dollar for something like that compared to a fresh market.
How common is it for growers to do like a pre-planting diagnostic sample?
Yeah, great question. We advise the growers to collect their soil samples in the fall to make decisions for the following planting season. That's because we know these nematodes are moving up and down in the soil profile, and it's easiest if they can sample relatively shallow, 8 to 10 inches deep in the fall, and get that information. It's easier for the nematode assay lab to extract and count when these populations are high. But it's getting more and more common that growers are sampling, which is a good thing. And generating this knowledge of what's going on from year to year in their field is very important so that they can make these long-term decisions. And especially with enterolobi, that's becoming more and more forefront in the mind of let's go collect samples or have our field scout collect samples.
Right.
Save my ugly question for dinner later, but maybe the softball question. You mentioned that 90% of sweet potatoes are this one variety.
Yes.
If you had a choice, what variety would you grow and why?
Yes. So 90% is that Covington variety. Covington has tolerance to Noetigae incognita, southern root knot, but is susceptible to the enterolobi. There are some other varieties. There's some African landraces that have excellent resistance to enterolobi. The problem is they don't have other market qualities that are acceptable in the U.S. market. The U.S. market wants an orange flesh, orange skin, relatively yay-sized sweet potato. These other ones are white skin, purple skin, very large, knotty potatoes. They grow well, but they won't be accepted by the market. Some of the purple sweet potatoes also seem to have some resistance we're finding. So I think they look really cool. That's the one I would grow.
Any other questions? No, not that. Thank you. Thank you. My account I'm doing it out. No,